All right. Sorry, guys. I am technologically inept, as you know, and I didn't realize that my 15 minutes was up because there's no countdown on this screencast. I have yet to figure that out, but hopefully I will get that resolved sooner rather than later. So where I got cut off, I told you that protists have three different variations of sexual life cycles, and the one that you're most familiar with is how animals reproduce, which is you end up with a diploid organism when you have two haploid cells that undergo fertilization, and you get your diploid zygote. And that's in all animals. Plants and some algae have actually quite a complicated life cycle, believe it or not. They reproduce by a mechanism called alternation of generations, and it's exactly what it means. It alternates between two generations of being a haploid individual and a diploid. And I will reiterate, haploid is where you have one set of chromosomes, and then diploid is you have two sets, one from a copy from mom and a copy from dad. So in the first stage of alternation of generations, the organism has both the haploid and diploid multicellular stages. Let's use a familiar example that you all know, and that's moss. You see moss a lot here in the Pacific Northwest when you're on hiking, and it might even be in your backyard, the lovely green carpet that we all like to rip off cement or when we're hiking, we rip it off trees. The moss has two stages that you might not be aware of. The first stage is called the sporophyte stage, which is the haploid stage. Maybe some of you are observant and have noticed there's these stalks and capsules that grow out of moss, and that is the sporophyte stage. And believe it or not, spores are reproduced in the sporophyte stage, hence the name. So these haploid spores go through a process called meiosis, and that's the same process that our sperm and egg cells go through. These haploid spores grow into a haploid multicellular organism known as a gametophyte. The gametophyte is the actual moss. So the moss that you pick off of other plants or step on is actually the gametophyte. And the gametophyte is the organism that produces the gametes by mitosis. These gametes that are reproduced undergo fertilization, and they produce a new multicellular diploid sporophyte, and the whole cycle begins again. So like I said, it alternates between two generations, your haploid and your diploid stage. So that's your second variation. So far we have two variations. We have the animal variation of sexual reproduction, and then we have the plants and some algae, which is alternation of generations. The third variation is the most unnatural way of thinking of how animals reproduce, and it's seen in fungi and some protists. So the only diploid stage of this cycle is the very beginning when the two gametes fuse to form a diploid zygote. So when the zygote is formed, meiosis occurs without a multicellular diploid offspring developing, which goes against everything that we've ever learned biologically. When we know from since sixth grade, when you started learning about meiosis and sexual reproduction, when a sperm and an egg fertilize, they make a diploid zygote. In this variation, meiosis occurs, but there's no diploid offspring developing. It basically reduces it, and it makes haploid cells that divide by mitosis, making a haploid individual. And this haploid individual can be either single-celled or a multicellular organism. And that would be your protist, because we know that fungi is eukaryotic and it has to be multicellular. Next, you have the haploid individual then produces gametes by mitosis. And again, that goes against all things that you learned. We know that most gametes are produced through meiosis. However, in this variation, gametes are produced by mitosis. And the last thing you need to remember is that the only diploid stage here is called the single-celled zygote, which is the first stage of this process. The big takeaway message that you need to know of all of these variations of sexual reproduction is that any cell, whether it's haploid or diploid, can undergo and divide by mitosis. So those are your three variations. I'm not on the exam. I'm not going to ask you too much detail about what happens in each one. I will. I might mention very basics, like what is alternation generations, what is the difference between a haploid, it goes between a haploid and diploid individual. Um, but you'll see more of this in plants. We end up really spending a lot of time on this topic. I mainly just want to mention it to you that protists can undergo all three of these variations. So here's a nice chart for you to look at, or illustration of the three mechanisms of fertilization. So here we have animals. You have your meiosis occurs to produce your sperm or your egg. These two gametes undergo fertilization that produce your diploid zygote, and then you get your pretty little animal. Here is a field mouse. The next is plants and some algae. This is alternation of generations. Here in alternation of generations, down here we started to talk about the sporophytes. These are your stalk and capsule. And then this is the gametophyte, which is the actual moss. So here, in the haploid multicellular organism, or the gametophyte stage, this is where your two sex cells are reproduced by meiosis. Fertilization takes place, and a zygote is formed to produce this this diploid sporophyte that produces the spores that undergo meiosis. So it goes between haploid and diploid multiple times. So it alternates between two generations. And then finally, we have the haploid unicellular or multicellular organism. So this is most fungi and some protists. Here, like I said, we have a zygote that forms, and usually that is the combination between a sperm and an egg undergoing fertilization and making a diploid organism. However, in this scenario, meiosis splits the zygote into a haploid organism, and then this organism undergoes mitosis, which gametes are formed, and fertilization takes place, and then you get your zygote. So those are your three variations. Lastly, we need to talk a little bit about evolutionary history of protists, which is really hard to discuss because, as we talked about, in the first mini lecture, that they are not monophyletic, meaning they do not arise from one common ancestor. So it's really hard to distinguish where they may have come from. But if you remember way back in September, we talked about endosymbiosis, or the endosymbiotic theory. And this is where we believe eukaryotic cells engulfed prokaryotic cells, and they lived in a symbiotic relationship, and their remains became their organelles. So, a parasitic cell engulfed maybe a mitochondria or chloroplast that instead of digesting it for food, they ended up living together in harmony, and then they ended up living as a system, and now chloroplasts make energy for plants, and mitochondria make ATP for animal cells. So this primary endosymbiosis is believed to happen twice. So there's your primary endosymbiosis and your secondary endosymbiosis. So the first is the aerobic prokaryote was engulfed. So remember, aerobic means we need oxygen. Mitochondria obviously undergo cellular respiration in the Krebs cycle, so we need O2 to undergo, to generate our adenosine triphosphate. This has led to the acquisition of our mitochondria, and all eukaryotic organisms we've studied so far have mitochondria. The second is your photosynthetic. Cyanobacterium was engulfed, and cyanobacterium do make their own energy through light energy, and they were engulfed, and these became plastids. These plastids eventually led to algae and finally plants. I told you way back in um, evolution lecture that algae is not a plant, it is a protist. They have very similar characteristics to plants, but they have some distinguishing characteristics that do not make them a said plant. One, hence being they lack a cuticle, which is all land or terrestrial plants have that, so they don't dehydrate through transpiration. Algae, as you know, has to be submerged in water. Again, all these organelles formed by primary endosymbiosis, meaning your mitochondria and plastids, and they have a double membrane. So if you were to cut open a chloroplast or mitochondria, they're both bean-shaped organelles. If you remember from IB Bio 1, inside those organelles, you have double membranes. So like the thylakoid, where your light independent and dependent reactions take place. And in mitochondria, that's where the Krebs cycle takes place in those inner membrane foldings. So you can see that those were once their own entity, and then they lived in an endosymbiotic relationship with the original cell. So the inner membrane of the cell represents the original prokaryotic. Here there are two possible origins of the outer membrane. So it may represent the engulfing of a vesicle, or it may be of bacterial origin. The engulfed organism was gram-negative with two bacterial membranes. We're not 100% sure what the end-all be-all answer is, because we know that in gram-negative bacteria, they have lipopolysaccharides in their plasma membrane, which we know that eukaryotic and prokaryotes do not have lipopolysaccharides. We do have some traces of peptoglycan and lipids. So, and then in some red algae and green algae, they show evidence of secondary endosymbiosis. In secondary symbiosis, a heterotrophic eukaryotic organism engulfs a second heterotrophic eukaryotic organism. These more modern organisms have plastids that are much more complicated with more than two membranes. So these are a little bit more sophisticated organisms that have predatory tendencies that already have engulfed mitochondria or chloroplast and are their own little entity. And they swallowed another heterotrophic organism for nutritional value, but they ended up living in a symbiotic relationship. Chlorochenophytes is the first lead example of this. Chlorochenophytes, you'll learn, that are a type of algae that we'll study, and they have plastids with four membranes. So this just goes to show you that there was an original, and then there's two inner membranes that they had to believe come from the original endosymbiosis. The third membrane is from the plasma membrane of ingested algae, and then the outermost is from the food vacuole that engulfed it. These cells also have a nucleomorph in between the two middle membranes that form from the former plasma membrane and the original plastid. A nucleomorph is a vegetal nucleus, meaning it looks like a nucleus. It probably functioned once as a nucleus, but it serves no purpose. It's kind of like our appendix, right? We don't eat massive amounts of cellulose anymore because we are obviously now, unfortunately, more carnivorous than we are herbivores. So over time, we have not needed our appendix to help us with digestion of cellulose. So it's become an organ that we can live with or without and still function 100% normally. So that's the same idea with a nucleomorph. So here we have our heterotrophic eukaryote. That's your primary endosymbiosis. It engulfs, the dark lines represent the cell and the membranes. One of these membranes was lost in red and green algae descents. So here they are. Secondary endosymbiosis occurs. This organism that's living with mitochondria or plastid, whatever it may be, In this case, we're talking plastids because we're talking about algae, and we know algae